I'd like to start my presentation on the prevention of chlorine-based fall caused by calcium chloride snow remover with carbonate-forming bacteria. When oversnowing occurs, it impedes with daily life of humans. Therefore, common snow removers such as calcium chloride are used. Although they are known to be toxic and bad for the environment, they are convenient, cheap, and effective. However, I have discovered a new problem that hasn't been anticipated. When the calcium chloride seeps into the ground, flows into the water cycle, and evaporates, chlorine gas may form in the atmosphere, causing chlorine ions to enter the water cycle. Previously, I have made an eco-friendly snow remover made from oyster shells. While it was effective, it did not match the snow melting efficiency of calcium chloride. Therefore, I set out to look for a solution to make the calcium chloride less harmful. After research, I found a bacteria that utilizes chlorine ions to form an outer shell of calcium carbonate for itself. I thought this could be a possible solution to the problem. Experimental design involves the following process. First, the new problems were identified, a solution was formed, and the logistics for the solution was tested. To clarify the experiment designs, there is an outer container with water inside, and the petri dish is on top of the water. The whole container is then sealed with a lid. The variables consist of control, which is nothing, direct, which is tested when variable is added into the petri dish directly, and indirect or side, which is added in the water on the side to test the indirect effects. First, 1 gram of calcium chloride was dissolved in 100 ml of distilled water to make a 1% calcium chloride solution. This solution was then taken to test the problems, which were on water evaporation, effect on growth of plants, phytoplankton, and soil bacteria, and the generation of chlorine gas. Then for the selection of calcium carbonate forming bacteria, a medium was made and sterilized in the autoclave, cooled at room temperature, and refrigerated until use. Then water is run through 4 different types of soil were smeared on the medium to select calcium carbonate forming bacteria. With the experiments for testing the logistics of the calcium carbonate forming bacteria, plant growth, phytoplankton growth, and the generation of chlorine gas was tested. The same procedures were used for these experiments, but just with the bacteria added. Now the overview of the results of the above analysis. First, there will be results for the confirmation of the problems, and then the logistics for the solution. When looking at figure 7, which is the increase-decrease of soil moisture content, while the control group shows a decrease in soil moisture content due to evaporation, which is why it shows as a negative, the direct group D and indirect group S, in which calcium chloride was administered directly and on the side, shows a comparatively high moisture content, which means there was an impediment in absorbing and evaporating water. Now for the soil bacteria in figure 8, when the calcium chloride was directly and indirectly administered to the soil, the absorbance of soil bacteria was significantly reduced compared to the control soil. There was also a significant difference in bacterial colonies. As seen in figure 9, the control group showed a variety and abundance in bacterial colonies. However, the indirect group and direct group shows a significantly decreased abundance of colonies. The calcium chloride also shows significant damages in phytoplankton growth. As shown in figure 10, while the control group shows significant amounts of phytoplankton, the indirect group and direct group shows little to almost no phytoplankton. Additionally, it also impeded with plant growth. In figure 11, the plants of the direct and indirect administrations were comparably less healthy and grown less than the control group. Not only visually were they damaged, but even when the absorbance measurements of the chlorophyll A and B were measured, the control group showed a significantly higher measurement than the direct or indirect group. The existence of chlorine gas was also confirmed when the water containing calcium chloride evaporated into a gas. When the condensed liquid on the lids were tested of chlorine content, as shown in figure 12, both the direct and indirect group showed a significant amount of chlorine ions. For the solution to this problem, a bacteria that utilizes chlorine ions was selected. As seen from the process on the left, the cell takes the chlorine ions and produces calcium carbonate, which forms on its outer shell. The cell was selected because the calcium chloride was proven to evaporate and precipitate containing chlorine ions. Now for the results after the bacteria was added. First, the plant growth was restored back to normal with the bacteria. Visually, when looking at figure 14, with before on the left and after on the right, while the direct D and the indirect S's groups plants struggled to grow before administration, the direct and indirect groups plants flourish as much as the control group after the adding of bacteria on the right. Next is soil moisture. On figure 15, the top is before and bottom is after bacterial administration. While the top shows a high moisture content for the direct and indirect group, which conveys a low evaporation rate, which impedes to the water cycle. The bottom shows a high evaporation rate, even for the direct and indirect groups. Next is the phytoplankton recovery. Figure 16 shows left as before and right as after addition of bacteria. Before, the phytoplankton is clearly struggling to live in the direct and indirect conditions. However, when looking at the right side after the bacteria administration, there are clearly more signs of phytoplankton, even in the direct and indirect administrations. The chlorine content in the fog also had decreased. In figure 17, the left half from each soil and seawater shows the initial chlorine content. As you can see, there are clear signs of chlorine in direct and indirect group. However, on the right side after the bacteria administration, the chlorine content either reduced to the same level as control or significantly decreased as shown by the lightness of the color. A clear new problem has been discovered, which is the possible produce of chlorine gas when the calcium chloride enters the water cycle. The damage that it can do was also confirmed. Therefore, the calcium carbonate producing bacteria, which is process is shown on the side, can work as a solution because it has shown to nullify damages done to plants, restore water evaporation in soil, showed recovery and growth effects on phytoplankton, and suppress chlorine gas from generating. Therefore, this poses as a plausible solution. For possible future studies, I want to make this study as a stepping stone for something bigger. For this to be applicable in real life, I want to figure out the exact amount that is best and the method of distribution. Additionally, I want to see if the calcium carbonate produced can benefit the environment in any way. Thank you.